Okay, welcome back to the study of virology. Now we are going to study about the structure of TMV. The expansion for TMV is tobacco mosaic virus. Why this virus is called the tobacco mosaic virus? This virus is attacking only the tobacco plant. You know very well what is a tobacco plant. The leaves are very broad and from the leaf only they are manufacturing the cigarette. That is tobacco plant. So in the tobacco plant, the viruses are attacking the tobacco plant. They are causing a disease called the mosaic disease in the leaf. What is that mosaic disease? We have to say. Normally, the leaf of any plant, if you take in the higher organisms, higher plants, the leaves will be broad. The leaves will be completely green because of the presence of a green color pigment called chlorophyll. Lower classes you might have studied. With the help of the chlorophyll, the green leaves can prepare food material by photosynthesis. So as a result of photosynthesis, starch will be prepared. And the starch will be stored in the plant and it is used by the plant. That is the purpose of the green color leaf. But once the virus is attacking the tobacco plant, the leaves of tobacco will become mosaic leaf. What is the meaning of mosaic leaf? The disease is also called a mosaic disease. The leaf is a very broad and green. But once the viruses enter in the leaf, various colored patches will appear everywhere, very close, like yellow color patches, brown color patches, orange color patches, black color, like that. So many colored patches will be formed fully. So as a result of which, the leaf cannot prepare any food by photosynthesis. So the plant cannot get sufficient food. At the end, the plant will die. That is actually the tobacco mosaic virus. Now the tobacco mosaic virus, what is the structure we have to study? Basically, the tobacco mosaic virus is having only two parts. One is the outer protective covering. You can see that yellow color. It is a thick protective covering. It is called the capsid. It is also called the protein coat. Then inside the protein coat at the center, there is a long coiled thread-like structure. This long coiled thread-like structure is present from end to end. It is called the RNA. So the tobacco mosaic virus is having RNA as a genetic material. Now this RNA, you can see that it is a long thread and it is kept in the form of coiled thread-like structure is going on. And outside that, the protein molecules are present. They are forming one layer completely all around. So we cannot see the RNA at all. So only the protein coat we can see outer. So that is called the capsid. Now the capsid, the protein coat, is having small blocks. That is small units are present. Each unit is called a capsomere. Now I am giving the spelling for that capsomere. C-A-P-S-O. Capsomere. M-E-R-E. Capsomere. Enormous number of capsomeres join together closely and form the capsid, the protein coat. This is actually the structure of the tobacco mosaic virus. It is very electron microscopic, very small, very difficult to see by, that is even in the electron microscope, high power microscope we can see this TMV. The next virus we are going to study, it is a bacteriophage. Already we have studied what is bacteriophage. Phage means virus. Bacterio means these viruses are attacking bacteria. That means they are very smaller than bacteria. Maybe 1,000 times smaller than bacteria. That is bacteriophage. Different type of bacteriophages are there. Different type. In that one type we are going to study T4 bacteriophage structure. What is the meaning of T4? The T stands for tadpole. Tadpole is the larva of frog, which is having the body shape like this. Anterior it will be bulging and the posterior only narrow tail will be present. More or less giving appearance like young fish it is giving. That is a tadpole. Now the same tadpole-like shape this virus is having. You see this is a virus, generally will take. Now this is a head which is broader, elongated and broader. It is giving appearance like the head of the tadpole. And it is having a narrow, long cylindrical path. It is like the tail. So the whole shape of the virus is giving appearance similar to that of the tadpole larva of a frog, so that we are calling it as a T virus. Tadpole shaped virus, the T virus. In the tadpole shaped virus, different divisions are there. T2, T3, T4. Now we are taking only T4, one example. That means it is a one type of tadpole shaped virus. That is, it is a bacteriophage and T4. What is the structure of T4 bacteriophage? Now this is the bacteriophage virus. What is the structure we have to see? It is a having anterior elongated broad portion. This broad portion is the head. In the broad portion at the outermost region, we can see the outer protective covering. It is called the protein coat. This protein coat is also called a capsid. We are also calling it as capsid. Inside the capsid, we can see DNA. The bacteriophage is having DNA. Normally, viruses which are attacking plants will have RNA, but this is an exception. It is having DNA. It is having single strand of DNA. But in the diagram, it is kept very simple, highly coiled form like a spring from end to end. Generally, if you take the DNA, imagine like that, by research we are doing. We are taking the DNA and we are stretching to become a long, single, stretched twine line. It will be 1,000 times longer than the actual length of the virus. So, what is the length of the virus? In that 1,000 times the DNA length will be present. That is the DNA of the bacteriophage, T4 bacteriophage. That's all about the head. So head is having only two components. Outer protective covering called the capsid, and in the single strand of DNA is present, which is kept in a highly coiled form. Now below the head there is a sheet of protein coat. A sheet of protein coat will be present. It will be flattened, circular flattened. It is called a collar. Collar means like neck. Even we are also having collar, that is neck. That is called a collar for the virus. Then below the collar, a long cylindrical part is present. A long cylindrical thread-like structure is present. Very, very narrow. It is not broad like this. Very narrow. This portion is called the tail. It is actually called the tail. But the tail is having only protein coat at the outer region, but inside it is a hollow cavity. One cavity is there. It is like a tube-like structure. So the outer covering of the tail is called the sheath. That is protein coat. Then at the bottom of the tail, if you see, there is a plate again. A thick plate is present with the hook-like structures. This thick plate is called the basal plate. The basal plate is called the basal plate. Now attached with the basal plate, about 6 or 7 long thread-like structures you can find all around. Very, very long thread-like, it's kept folded like this. They are called tail fibers or tail filament. So here if you take the tail, at the anterior end of the tail, that is below the head, what is present? Collar. At the lower end of the tail, what is present? Basal plate. Now this is actually the structure. But if you take starting from the collar up to the basal plate and the tail fiber, everything is made up of protein. Everything is made up of protein coat. That is the structure of the T4 bacteriophage. Okay, this is the structure. Now we are coming to the life cycle of T4 bacteriophage. Now multiplication. Multiplication means how they are reproducing. That is called. Now when they are reproducing, whenever they are entering into the living cell, they can multiply. So multiplication, or otherwise called the life cycle. How it is keep on multiplying, that is complete thing is called what? Life cycle of bacteriophage. What is a bacteriophage? Just now we have seen one diagram, T4 bacteriophage. That is. Now in the life cycle, in the multiplication, there are two different types of it. One is called the Lytic cycle. One is called the Lytic cycle. Now this is one type of reproduction, multiplication. It is immediate multiplication. So there is a cycle means life cycle. What is the meaning of Lytic? I will not tell now. At the end, after completing this multiplication, I will tell what is the Lytic. Why it is called the Lytic cycle. Now, already we have studied it is the life cycle of bacteriophage. That means it is a virus which is going to attack only the bacteria. That is called the bacteriophage. Now, this is a bacterial cell. Now, this is the outer cell wall. It is having cytoplasm, the yellow color, cytoplasm. It is having circular bacterial genome, genetic material. Genome means genetic material. It is having circular genome. Then, imagine that the virus is trying to enter into the bacterial cell and it is going to multiply. So that is the whole thing is called the life cycle. The life cycle has 5 stages. One is called the adsorption. Adsorption is also called the attachment. It is also called the attachment, adsorption. Second stage is called the penetration. Something of the virus is penetrating into the bacterial cell. That is called the penetration. Synthesis. The viral particles are synthesized, produced in the bacterial cell, host cell. What are the viral particles? Only 2, protein coat and the nucleic acid. Okay, synthesis. It is also called the synthesis. Fourth one is assembly. That means viral particles are assembled and forming virus structure. It is also called the maturation. And the last one is release. The viruses are coming out of the bacterial cell. That is called. So 5 stages of adsorption, penetration, synthesis, assembly and maturation and release. Now we are going to say this adsorption. Not absorption, it is adsorption, attachment. Now this is the bacterial cell I told you, it is having all its components. Now one virus, bacteriophage is coming and attaching on the outer surface, anywhere, here, anywhere it will come and attach by chance. It is by chance. At the time when the virus is having contact with the bacterial cell, always the basal plate will be touching the wall of the bacterial cell, basal plate. Then the basal fibers, tail fibers are there, tail filaments are there. They are holding the outer surface of the bacterial cell, holding very strongly, so that the virus cannot be separated from the wall of the bacterial cell. Now this attachment is called adsorption. Now once the virus is attached, only the protein coat is attaching, basal plate and filaments are attaching. Now from the neck is having a cavity. One enzyme is coming to the wall of the bacterial cell. One enzyme is coming and touching. The enzyme is present in the virus. So what is the name of the enzyme? It is called lysozyme. The name of the enzyme is lysozyme. Now the lysozyme enzyme is here. It is slowly coming and touching the wall and the wall of the bacterial cell and making a passage, making a hole. Now once the hole is formed, the adsorption is over, attachment is over. Now we are coming to penetration. Now already the bacterial cell wall is having a hole. Now the virus is attaching on that hole. Now through the hole, the DNA what is present in the virus. Now it is called a phage DNA, virus DNA. Now the DNA is present in the form of long thread. Now it is passing through the tail, because the tail is a tube-like structure, and it is passing through the hole of the wall of the bacterial cell. And only the DNA of the virus alone is entering into the cytoplasm. So the phage DNA, viral DNA is entering into the cytoplasm of the bacterial cell. This is called the penetration. The DNA penetrate and enter into the cytoplasm. Okay, that is called the penetration. Now in all living organism, one property is there. Now this is the host cell. Virus is the parasite. Virus is the parasite. Whenever the DNA of the parasite is entering inside, the DNA, for example in this, the phage DNA is a foreign DNA. Why we are calling it as foreign DNA? Now the bacterial cell is having its own DNA. That is called the original DNA, own DNA. But this DNA is not belonging to bacterial cell. It is coming from the virus. So it is, as far as the bacterial cell is concerned, it is coming from outside. So it is foreign DNA. Now whenever the foreign DNA enters into the host cell, the foreign DNA will be functional and the bacterial DNA will become inactive. The bacterial DNA, so far it was working. But once viral DNA enters, this DNA is becoming inactive. Now what the phage DNA is doing, it is making its own activity. Now it is having information. What is that? Formation of protein coat and a formation of the RNA of the virus, DNA of the virus. By using the cytoplasm, the protein coat and the RNA will be produced in the next stage that we are going to see in the synthesis. Synthesis means production. Synthesis means production, manufacturing. Now the viral DNA has come. Now the viral DNA wants to multiply. Now it is one DNA strand. It wants to multiply into 100 or 200 or 300. So it is not having any other material. So it is making use of the cytoplasm and DNA, converting everything into raw material, and slowly the entire inner content of the bacterial cell will be changed into protein coat. You see that lot of protein coat pieces will be formed, and also the DNA slices will be formed. The DNA strands will be formed. So this production of protein coat and the production of viral DNA, phage DNA, now is called synthesis. So slowly the cytoplasm and DNA of the bacteria cell will be used. So the cell is becoming more and more empty, and a lot of viral particles will be produced. That is called a synthesis. Now imagine that enormous number of DNA of the virus will be multiplied. Enormous number of protein coats are also formed. Then we are coming to assembly. We know very well what is assembly. For example, we are taking mobile. We are dismantling it. We are removing the cover. We are removing the battery. We are removing the SIM, everything. That is called a dismantling. Then after that, what we are doing, we can assemble it. Once again, we can keep everything in the proper position. That is called assembling. Similarly, here also assembling. Now, during this assembly, inside the bacterial cell, what will happen? Now, the DNA of the virus will enter into the protein coat. Now, once the DNA enters into the protein coat, the head of the virus will be formed, the tail of the virus will be formed, and also the filaments will be formed. Now that means one actual small virus will be produced. It is T4 bacteriophage. Like that, many copies of bacteriophages will be produced. Many copies. So the end of this synthesis, that is at the end of maturation, if you take in one bacterial cell, 200 to 300 T4 bacteriophages will be produced. Now, once the assemblage and maturation is over, the bacterial cell is dead already. It is not having anything inside. It is having only outer cell wall. So only viruses will be filled in inside. Then we are coming to the last one, release. Now once the viruses are well developed, very well developed, then all the viruses are ready to come out. At that time, any one place in the wall of the virus, bacterial cell, anyone, here or here, anywhere, the wall of the bacterial cell will break. Now breakage of this wall. Now through the breakage, all the viruses are released to the surrounding. That is called a release. So one virus is entering into, not actually the full virus is entering, only DNA is entering into the bacterial cell. But 200 to 300 viruses will be produced. It is multiplication, it is a reproduction, it is a living activity. Now this is actually called the Lysogenic cycle, multiplication of the bacteriophage virus. Why we are calling it as lytic? Lytic means breaking. Now see the last division, that is release. At that time, the wall of the bacterial cell is breaking to release the viruses out. Because of the breakage, we are calling it as lytic. Lytic means breakage. Lytic cycle. Now we are going to see the second method of reproduction in bacteriophage. It is called the Lysogenic cycle. Now we are going to see Lysogenic cycle. It is another method of multiplication of the virus. But here also I am not going to tell what is the meaning of lysogenic now. Only at the end I can explain about it. What is the meaning of lysogenic? Here cycle means multiplication, reproduction, life cycle. Anyway we can tell. Now the basic reproduction will be the same like previous one. Now this is a bacterial cell. It is a host cell. Now adsorption takes place. That means one bacteriophage virus is coming and attaching. The basal plate is attaching. Even the tail filaments are holding the wall very, very tightly. Then penetration takes place, just like second step, penetration. That is the enzyme is coming here and dissolving the wall. So a hole is formed. So the DNA is entering into the cytoplasm of the bacterial cell. Now this is already bacterial DNA. Now viral DNA is entering inside. Now the viral DNA is a foreign DNA for the bacterial cell is concerned. It is foreign DNA. Now after entering it in this lysogenic cycle, what will happen? This is the viral DNA. It will form a circular arrangement like this. Now this is the bacterial DNA. Already it is present here. But in this method of multiplication, what will happen? The viral DNA is slowly moving and it is associated with the bacterial DNA, joining with the bacterial DNA. Now this is called the integration of DNA of virus and the bacteria. Integration, DNA integration. Now once they are integrated, they present together, nothing will happen to the bacteria. The bacterial cell is a leading normal life. It is having its own life, it is growing. Now it is attaining maturity. Now this is the matured bacterial cell. Along with that, even that integrated DNA will be present. This is the bacterial DNA and this is the associated viral DNA. Both will be present. Now the bacterial cell after maturation, it is ready to multiply. It is ready to multiply. The bacterial cell is multiplying by binary fission. In lower class we have studied what is binary fission. One cell split off into two cells. The same way what will happen in this, that bacterial DNA and the viral DNA together we can call as integrated DNA. What it will happen? The integrated DNA will duplicate. One integrated DNA will become two. That means both are exactly same copies. So two integrated DNA will become. One will move to this end, one will come to this end. So here one integrated DNA, here one integrated DNA. Now once the two integrated DNA are produced, then the cell wall will constrict at the center. Now you can see this constriction. And the constriction is growing deeper and deeper between the two integrated DNA, and finally the cell is divided into two. Now here it will be completely separated. So one bacterial cell is having integrated DNA, another bacterial cell is also having integrated DNA. What is that integrated in the daughter cell? Now the DNA of the bacterial cell will be there, DNA of virus will also be present. Now like that, the two young bacterial cells again will grow with the integrated DNA. Again they attain maturity, again they keep on multiplying. So the bacterial cell is multiplying. So after enormous number of multiplication of bacterial cells, at any time when the condition is becoming favorable, the DNA of virus will be separated. Maybe after many multiplication, the DNA of the virus will be separated. And after that, it is taking domination and it is making use of the DNA of the bacterial cell as well as the DNA of the cytoplasm of the bacterial cell, just like Lysogenic cycle, and making them to become protein coat and the DNA of the virus. Enormous copies will be produced. Then assemblage will be there. Then the bacterial cell will break open and all the viruses will come out. So the difference between the previous multiplication and now. Previous, once the bacteriophage DNA enters inside, it is keep on multiplying and the spoiling the bacterial cell and coming out. Immediate multiplication. But in the second time, the viral DNA is present, very inactive for a long time. But when the condition is becoming favorable, it is separated from the bacterial DNA and multiplying. This is called a lysogenic, allowing the host to grow and multiply, but any time it will be separated and it will multiply. The virus will multiply. That is called not lysogenic cycle. Now this is the second method of reproduction, but it is a slower method. It will take long time for that. So, but both method will be possible in the bacteriophage multiplication. Now we are coming to the related terminologies connected with this virology. A number of related terminologies are present. What are they? Now the first one, virion. Already in the introduction of the topic I told, virion means poison. From that term virion only, it is a Latin term. The virus is derived. So it is intact. Intact means full. Virus particle. It is intact virus particle outside the host cell. That means it is not in the host. It is present outside, either in water or in the soil or in the air. But the whole component of the virus is called a virion. That is protein code plus RNA or DNA. Now, viroid. We can pronounce it this way, viroid or viroid. Anyway, we can pronounce it, both are correct. Now, viroid means ssRNA without capsid. Protein code will not be there. Only the RNA we are taking. Single strand RNA. It is called a viroid. The third terminology is virusoid. Virusoid means a double strand RNA. Two strands of RNA. Okay, double strand RNA, circular RNA. Even sometimes the RNA will be circular. Linked with the large viral RNA. Sometimes it is linked with the large viral RNA. So that is called a virusoid. Prions. Okay, prions, P-R-I-O-N-S, prions. Infective proteinaceous particles. Now this is very, very important. Normally DNA or RNA will be infective substance. The protein coat will never enter into the host cell. But here, RNA or DNA will not enter. Only the proteinaceous particles will enter. Infective proteinaceous particles causing fatal means causing death. Fatal degenerative diseases in central nervous system for higher animals. In the higher animals, the central nervous system is brain and spinal cord. These virus particles, what is that? Proteinaceous particles are entering into the host cell and causing degenerative damage, that is diseases in central nervous system. Central nervous system, brain will be affected by virus. The spinal cord will be affected, as a result of which lot of problem will be there in the host, and finally the death will occur. That is actually called. Example for this, Creutzfeldt. Now Creutzfeldt Jakob disease. It is one type of CNS disease. Now CJD, that is abbreviation for Creutzfeldt Jakob disease. The second one is bovine spongiform encephalopathy. Bovine spongiform encephalopathy. Now here encephalo means brain. Problem in the brain, encephalopathy. Now this is bovine spongiform encephalopathy, BSE. It is mad cow disease. When the infection is taking place in the cow, the brain is very much affected, as a result of which the cow is becoming mad, restless, and finally it will die. That is. Then, scropic disease of sheep. Another one disease called as scropic disease. Now that is also caused by this. E. Cyanophages. Now this is one important thing, cyanophages. What is the meaning of cyanophages? Phages means bacteria. Cyano means blue-green algae. Not pure algae, blue-green algae. They are not like algae, they are not like bacteria, in between. That is called the cyano, blue-green algae. Cyanophages means these are the viruses which are attacking blue-green algae. Now you can see viruses attacking blue-green algae. So this one terminology is a new term. Mycophages. Here phages you know very well, viruses. Myco means fungi, non-green plants, fungi like mushrooms. Now that is, these viruses are attacking fungi. That is called the mycophages, viruses attacking fungi. The last one, the last part of the lesson, we are coming to viral diseases. So viruses can attack animals, viruses can attack plants, even viruses can attack human, because we are special animal group, so humans. Even viruses can attack bacteria, viruses can attack other type of microorganisms that we are not taking. The viruses, when they are attacking plants, plants are developing some diseases. Whenever the viruses are attacking animals, animals are developing lot of diseases, animal diseases. And the human beings, when the viruses are attacking human body, the human beings are getting lot of diseases. Now we are going to see what are the plant diseases. Now already mosaic disease we have studied. For the tobacco I told, the leaves will have different colored patches like mosaic type. That is called mosaic disease. If the mosaic disease is occurring in tobacco, it is called tobacco mosaic disease. Cauliflower mosaic disease. Sugarcane mosaic disease. Even in the sugarcane leaf, the long green leaf, different colored patches will appear. I will take again here. Now, cucumber mosaic virus. In the cucumber fruit, in the cucumber plant, the leaves will be affected. Then tomato mosaic disease. Even these are all the mosaic diseases. Apart from that, potato leaf roll. In the potato plant, the leaves will be little longer. They will be present above the soil. Potato is the underground stem, but leaves will be above the ground. But what will happen? The long leaf will roll completely, so that the leaf cannot get sufficient amount of sunlight, and the leaf will not prepare food sufficiently. Then bunchy top of banana. Now we know very well banana plant. The leaves will be very long, more than 1 meter. They will be long and folding down. But once the virus is attacking this, all the leaves will be clumped with each other, will join together at the top. They are becoming bushy-like. So the leaves cannot stretch completely, the leaves cannot get sufficient sunlight, so the plant cannot prepare sufficient food, the plant will die. That is called the bunchy top disease of banana or bunchy top of banana. Leaf curl of papaya. So in the papaya plant, leaf curl. Curl means folding. The leaves are folding, even papaya leaf will be broad, but the leaves will be completely curled, as a result of which food will not be produced. Vein clearing of lady's finger. Lady's finger is a vegetable in the plant, in the leaves. Leaves are having veins, midrib, veins, side veins and all will be present. But what will happen? Vein clearing. All the veins will be blocked by the viruses, so water cannot be moving, food cannot be moving, so complete problem will be there. So the plant will die immediately. Rice tungro disease. Even in this rice tungro disease, a rice plant means what? Paddy plant. Now in the rice plant, leaves will be narrow and long, but it will be normally green. But here also, yellow color patches will be formed on the outer surface. That is called rice tungro disease. The other two already we have studied as a mosaic disease. All these diseases are plant diseases. Now we are coming to animal diseases. Even some important, economically important animals are affected by viruses. Fishes, they are economically important. They are affected. Cattle will be affected. Sheep and goat, even pet animals will be affected. Like that, many animals will be affected by the viruses. So they are causing diseases called animal diseases. What are the animal diseases? The first one is foot and mouth disease of cattle, like cow, ox, everything. They are affected by this. Very commonly, they are affected by this viral disease. That is foot and mouth disease. What is the foot and mouth disease? So the foot, each leg is having two divisions. Okay, that is there at the gap. The viral infection will be there. So much irritation and much damages will be there in the place. Sometimes blood will come out from that. That is called a foot disease. Whenever the foot disease is there, automatically mouth disease will be there. The tongue will have lot of patches. And also from that, so the animal cannot take any food. The animal cannot drink even water. So the animal become very weak and die. That is foot and mouth disease. So when the animal is stretching the tongue, lot of brown or black color patches will be present. Then rabies of dog. Now this is rabies virus. Can enter into the dog. Normally present in the dog, every dog. And they are present in the salivary glands of the dog. They keep on multiplying. And whenever the dog is spitting the saliva will be automatically dropping down. In the saliva of the dog, enormous number of viruses will be present. But when the viruses are keep on multiplying and becoming extremely more in number, then the dog becomes mad dog. That is called a rabies dog. We are calling rabid dog or rabies dog. That is rabies of dog. Then the third one is encephalomyelitis of horse. Even human beings are also affected. That is a horse animal, you know. Now already told you encephalon means brain. Then myelitis means the brain is covered by some membranes. There the virus infection will be there, so brain will be affected. So the horse brain will be affected. It is called encephalomyelitis. So it will become restless and it will die. Maybe within a few days the animal will die. Then human diseases. Whenever the viruses are entering into our body, many diseases are caused. That is called, they are called viral diseases. Now we will take the first one, common cold. It is a respiratory disease. Now that is viral disease. Sometimes throat will be infected, running nose will be there, then the lungs will be affected finally. That is called a common cold. It is caused by common cold viruses. Then another one is the hepatitis B. It is a disease. It is a liver infection in the liver. The liver is affected. Hepatum is the liver. Now the hepatitis B virus is there. It is called a hepatitis B virus. Whenever that virus enters into the body, it is settling in the. As a result of which, the people are affected by jaundice. That is called a hepatitis B disease. Cancer, anywhere in the body, cancer will be produced. Cancer is caused by so many factors. Radiation can cause cancer. Chemicals can cause cancer. But even viruses can cause cancer. Some viruses are there. They are very, very specific. When they happen to enter into our body, they are causing cancer either in the brain or in the throat or in the lungs or in the stomach or in the intestine. Anywhere cancer will be produced by some viruses. The fourth one is the SARS. It is a type of disease. Now it is an expansion. Everything must be catered. S-A-R-S. Maybe about 1 decade or 2.5 decades before, the SARS was very, very famous, like how Corona is very famous now. Like the SARS was once upon a time, but it was controlled easily. Severe acute respiratory syndrome. Syndrome means symptom, disease. Respiratory syndrome, respiratory symptom, severe, acute, severely affected. Acute means quickly the individual is affected by the respiratory disease. That is by SARS. SARS virus is causing that. That virus is called SARS-V, SARS virus. Another one is AIDS. There is a disease. AIDS is a disease. What is the expansion for this? Acquired immunodeficiency syndrome. That is AIDS. Now that AIDS is caused by a type of virus called HIV, human immunodeficiency virus. This virus is reducing the immunity level of human. As a result of which we are getting immunodeficiency. Once immunodeficiency is there, other type of microorganism will attack the person and the person will die. That is AIDS. Next one is rabies. The rabies is a disease, viral disease caused by dog bite. Not only dog, but other animals are also present. Simply we can say dog bite. Whenever the dog is biting from the saliva, the virus will go directly into our blood. Now once our viruses enter into our blood, they grow, multiply. After that, it is causing a disease called rabies, which is affecting the brain, which is affecting the muscles of the respiratory system. So we cannot respire, we cannot keep our mind in a proper way. So death is the cause. It is very, very deadly. Mumps, a type of pox. Polio. Now the polio, if you take some paralysis, it is causing paralysis. Polio is caused by polio viruses, but now it is eradicated. Almost maximum it is eradicated. Now because vaccine is there, polio vaccine, polio drops, everything is there. So now it is not causing any person. It is not affecting any person, but the polio is caused by polio virus. If it is attacking, the hand nerves will be affected, legs nerves will be affected, as a result of which hands and leg development will become abnormal and paralysis will be caused. Chikungunya. Now the viruses will attack the joints of the wrist, knees, elbow, everywhere. As a result of it, joint degeneration will be there, severe problem will be there, pain will be there in the joints. Smallpox, we know that is also eradicated in our country. It is one type of pox which will attack the face. The face will become so ugly. So it is also deadly. Chickenpox, very commonly it is affecting, but seriousness will not be there. So we can easily recover naturally by chickenpox. It is caused by chickenpox virus. Smallpox is caused by smallpox virus. Measles, it is also the type of pox which is forming lot of blisters over the skin. Now all these things are some common human viral diseases. Now that's we have completed diseases, viral diseases. Now by this we are finishing the first chapter in biobotany. Previously introduction, now first chapter virology. By this we have completed. Thank you. God bless you.